Good morning to all. I am Dr. K. Sundaravadivu, Assistant Professor, Department of Biochemistry, Sri Adi Sundanari Women's College, Kambam. Let us see about DNA replication in prokaryotes. These are the contents going to be taken in this session, and briefly we can see about DNA replication. Before going to enter into the topic, we should know about what is meant by molecular biology. It is a branch of biology that deals with the structure and function of the macromolecules such as proteins and nucleic acids. Next is, what is the central dogma of molecular biology? That means flow of genetic information from DNA is converted into functional product. That is, one molecule of DNA is converted into another molecule of DNA. The process is called as replication. A DNA which converted into RNA, this process is called as transcription. And RNA is converted into protein is called as translation. At the same time, RNA is converted into DNA, it is called as reverse transcription. Okay, till now we know about definition of molecular biology and central dogma of molecular biology. Next, coming to the point, what is DNA replication and why should we know about DNA replication? As a biochemistry students, you should know about it because it is the biological process occurs in all living organisms and it acts as the most essential part for biological inheritance. In DNA replication, two identical replicas are produced. Replicas means same copy of DNA. That means in replication, a character transferred from one generation to next generation. How it does transfer? Because the structure of DNA is double helix and it has two complementary strands. That means which composed of two linear strands that runs as anti-parallel each other. In DNA, there are four types of nucleotides such as adenine, guanine, cytosine and thymine. These nucleotides are responsible for the transfer of information from one generation to next one. In the process of replication, the two strands are separated. How the separation occurred? What is the point of replication? That means a sequence found on the chromosomal DNA called origin of replication, the point at which the DNA opens up. In this point, the DNA replication gets started. During opening, each strand acts as a template for the production of new DNA strand. The DNA replication occurs in three types of mode. They are conservative, semi-conservative and dispersive. These mode of replications were described by scientists, but among these three types, semi-conservative mode of replication is occurred in all living organisms. Because in conservative mode of replication, entirely new molecule is synthesized from a new template. In semi-conservative mode of replication, each new molecule consists of one newly synthesized strand and one template strand. In dispersive model, new molecules are made of segments of new and old DNA. So among these three types, semi-conservative mode of replications are experimentally proved and accepted. In this DNA replication process, lot of enzymes are involved to carry out the process, such as DNA helicase, single-stranded binding proteins, topoisomerase, primase, DNA polymerase 3, RNA primers, and DNA ligase. Next, what is the role of enzymes? DNA helicase, which opens the DNA strand at the replication fork, and single-stranded binding proteins, which coated the DNA around the replication fork to prevent rewinding of the DNA. And the function of topoisomerase is which prevent the replication fork to prevent supercoiling. The primase, which synthesize the RNA primers complementary to the DNA strand. And the DNA polymerase 3, which extends the primers, adding on to the 3' end to make the bulk of the new DNA. And RNA primers, which removed and replaced with DNA by DNA polymerase 1. Then the gaps between DNA fragments are sealed by DNA ligase. Okay, next is, what are the steps involved in DNA replication? There are 3 steps: initiation, elongation, and termination.
இப்போ நான் பாஸ் தான் பண்ணிக்க இது என்ன பண்ண
பாஸ்